Hello, my fellow Everthinkers. In today's episode of our podcast, we are really pleased to be joined by Professor Anna Salvati. Over the next hour and a half, give or take, you'll hear a fascinating description of what nanomedicine is and in which direction it is evolving from one of the leading contributors in the field. Today we cover things such as exploring the meaning of the pharmacological buzzword that is nanomedicine and explaining how paradoxically it is a scaling up of standard delivery drug methods. And the things that nanomedicine aims to optimize in pharmaceutical therapy and the complexities of how nanoparticles act once they are in our bodies. Zed, what do you think of this podcast? I thought it was really fascinating.
It was fascinating, Oscar. I really was. I was intrigued by the by the depth and the complexity of the nanoscale, about how particles and blood particles and the drug particles could come together in the nanoscale and interact and introduce this amount of complexity, and how we could actually understand and approach this complexity to better use it and utilize it to our advantage in combating certain diseases. So, fascinating interview.
Exactly. You're going to hear us probably struggling a little bit to understand certain concepts because we stepped into the interview not really fully understanding certain things. So hopefully you'll enjoy that little journey we go on together. And yeah, hopefully you understand it as well. But it was a really good, really energetic, fun podcast. So that's all from us for now. And I hope you enjoy it. And thank you so much for listening. Professor Salvati, welcome to our program. We would first like to ask you to describe yourself. Tell us a little bit about yourself and I guess your journey in science.
Well, first of all, thank you for the invitation, and it's nice to be here. I'm originally from Italy, and I'm a professor in nanomedicine, and I'm here in Groningen University from 2014, almost 10 years now. And I work in the Department of Nanomedicine and Drug Targeting at the Pharmacy Institute. I am a biologist by training. So at the university, I studied biology at the University of Florence in Italy. I always wanted to understand how cell works, how life works. So it was really for me for understanding how alive cells works, what is the difference between a plant, an animal, microorganism, and really understanding the mechanism behind living matters, if you want. And later I've done a PhD in physical chemistry, and I started to get into the topic of drug delivery, how the drugs, medicines are transported in the body. From there, I did the PhD in physical chemistry, working on liposomes, lipid vesicles to transport DNA in that project. And after 3 years of PhD, I started a postdoc. For that, I went to Ireland, Dublin University, and there I went back a bit to biology. So I started to really work on using still drug carriers, drug delivery systems, but trying to understand how they interact with the cells, if they enter, where do they go, with the idea of, again, understanding the basic mechanism behind these interactions. Later in 2014, I moved to the Groningen University. I was awarded a Rosalind Franklin Fellowship to start my independent lab. So I moved here in Groningen, and now I am at the Pharmacy Institute since then.
Fantastic. And to move back a little bit, I guess what inspired you to go down that physical chemistry route that then led you to nanomedicine? What was the idea behind that? Because, you said that, I guess, biology as a concept and understanding living things and how they interact was kind of the initial thought.
Yes.
The physical chemistry is a little bit of a switch, in my opinion.
Yeah, absolutely. And I think that's a bit of personality as well, I guess, in the sense that curiosity and opportunities in life. So in the biology, we have a lot of basic chemistry, physics, mathematics, and then we had histology, human anatomy, molecular biology, genetics, physiology, a very broad spectrum of courses with this common theme in the end to understand how life works, how living organisms function. And one of the courses I did was physical chemistry, so a rather fundamental course, but I really loved the topic and my professor at the time asked me if I wanted to do the research project for my master degree with him. So I accepted that because I really liked that topic. And that brought me to the first experience with drug delivery. So my master project within biology was on making liposomes, so lipid vesicles that we use to transport DNA if you want to deliver genes inside cells. And it was mainly on the formulations of these vesicles, of these, nowadays we call it nanomedicines. When I did my master's and my PhD, perhaps we were calling them, referring to these objects, to this technology as drug delivery systems. So I did 1 year of research on preparing liposomes and how best to encapsulate DNA, how changing the lipid compositions to have the best interactions with DNA and transport the DNA. So this was mainly the physicochemical preparations and characterization of the formulation, if you want. Well, later, already with my postdoc and now in my work, what I'm doing is also testing that on cells to understand how by modulating the material properties, the drug carrier properties, I can affect the behavior with the cells so that I can also later use it for drug delivery, in fact.
Okay. So I'm thinking maybe, was the field at that time well established, or was it still growing into the field of nanomedicine that we are knowing today?
Yeah, definitely when I started my master project and later my PhD, we were calling about drug delivery systems, but it was only more recently that perhaps we switched the name to nanomedicine to stress these factors of the size of the materials that we're talking about. But drug delivery has been a topic of research already from many decades. You have to think the first nanomedicine, the first drug delivery system that was approved for clinical use was Doxil in 1995, which is a liposome that transports the chemotherapeutic drug doxorubicin. So that was already approved for clinical use in 1995. So the research to arrive to that approval started decades before. So it's already several decades of research into drug delivery. And now, since then, there have been a lot of other drug delivery systems, nanomedicines that have been approved, up to very recently the RNA vaccine against COVID-19.
Okay. And you mentioned this multinational, I guess, experience where
Yes.
you went from Florence in Italy. I know that you spent some time in Sweden as well, and then you moved to Ireland as well. How, I guess, did this experience shape you? Maybe how did like the mentors within maybe all these departments also guide you in your research? What can you tell us about that?
Yeah, that was quite an opportunity after another that I had. I started in Italy and did my study there, and also my PhD started in Italy in the same university where I did my master project. But then, exactly because I was coming from biology, but I switched to a PhD in physical chemistry, I've always been interested in this interdisciplinarity and topics that are at the intersections between different disciplines. But I felt that I needed to get stronger on the physical chemistry side. So I wanted to do an experience abroad. And then when I went in Sweden, so within the PhD, I went for a period in Sweden in a physical chemistry department at Lund University to embed myself even more in physical chemistry and in the end, I stayed there half of my PhD, and from there I moved to Dublin. But yes, so visiting different laboratories is always an incredible experience. Living in different countries at a personal level, it's a very enriching experience, but also for research. When I started the postdoc and then moving again here in the Netherlands, every time you move, you find yourself in a different research environment, and it has been an opportunity to being exposed to completely new expertise around me, new topics of research, and adapt to new environments. So definitely something very enriching, both from the personal point of view, from myself, and also as a researcher.
So is science actually international in a way, or is it more? Or did you notice that basically every region has its own way of thinking of science, or conducting science maybe, or coming up with ideas?
I think there is the way you do research and you do science. There is a lot that is shared regardless where you are. But definitely working in wherever I've been, there was a rather international environment, and so you anyway are also in contact with colleagues with different backgrounds, with different, not only different expertise, because we are if you work in an interdisciplinary field, also in Dublin, I don't know, I had the chemist together with biologists, together with physical chemists and people with more medical background. So not only you have an interdisciplinarity of discipline and expertise, but also cultures when you have people that come from different countries. But in the end, research is quite a shared
effort, basically.
Yes.
Yeah, because it's also interesting that you switched fields, and I can reflect on my time and probably on Oscar's time as a student that we think that if you do, for example, pharmacy, if you do a PhD in, for example, pharmacokinetics, you're going to stick to pharmacokinetics, but you have basically switched fields, and you just had the very strong case of follow your passion, right?
Yes. I don't know if the definitely switching from biology to physical chemistry was quite a switch. But then I went back to the biology with experience from the physical chemistry side. So it really depends in the end for the research that I'm doing now in drug delivery, in nanomedicine. I think you really need to have the capacity to both talk with the people that are on the chemistry side, that design the material for drug delivery purpose, and understanding the biology, how it works at the cellular level, and what does it mean that they enter, that they are internalized by the cells. So you really need to be able to talk different languages and perhaps, for instance, even if it's not me that do hardcore material chemistry for the design of certain materials, I need to be able to speak with those colleagues that do that, and as well as perhaps a clinician that has a need that needs to solve a problem and requires a drug delivery strategy. So, when you work in this kind of field, it's there are so many different aspects that it's good that you are exposed to this different expertise and are able to talk to each other. So when I switched to physical chemistry, I needed to embed myself even more in physical chemistry coming from the biology background. But then already when I moved to Dublin for my postdoc, I started again to work with cells and adding these materials to cells. So if my PhD was about designing the material, characterize the properties, the size, the charge, how drugs are loaded inside, how can I improve the amount of drugs that I put inside the drug carrier and this kind of aspects, then with that experience, I went to Dublin and I started to test these things more and more on cells to understand how the material properties affect what happens with the cells, for instance.
So you basically integrated the science or the knowledge from the physical chemistry that you got throughout your PhD with more of the biological...
From my background studies, yes.
And then your postdoc.
Yes.
So it seems to me that the borders in science, or the borders between degrees that we now know as students from biology, physics, chemistry, kind of...
They're changing, I think. They're mixing more and more. I mean,
there are clear...
and I guess pharmacy... Sorry, in that sense, pharmacy is a very interdisciplinary topic, I think, because if you think about the pharmacy program and what you study in the pharmacy, you go from chemistry and the design to drug design to understanding how cell works, understanding disease, understanding pathology. Actually, also in my biology studies, I told you I went... Also, it was a very interdisciplinary topic. I had some pharmacology exam. Those were some of the last year exams. And for me, in fact, it was beautiful because at that point, you understand a bit of the chemistry. You understand what the disease comes from, what is the mechanism behind the disease. So what is the molecular target, where your drug, your active molecules needs to intervene, what is the problem that needs to be fixed. If you know that how the certain molecule is malfunctioning, you know what happens in the cell, at the cell level and at the tissue. You can connect it to the pathology of the disease. And then you can think how to fix it and the pharmacology aspects, in fact. So when you arrive to think at how to treat a disease, you're really and what is the molecule that you need to use and how to transport it as well. You're really putting together all these different aspects from the design of the molecule that you need as an active therapeutic ingredient to what the disease is, what is causing the disease, and how to fix the problem. So pharmacy is a very interdisciplinary topic where you need knowledge of very different disciplines. Absolutely.
Yeah, absolutely. Like the sheer complexity of all the fields that we have would, I think, in the future would mean that we need scientists who can communicate throughout different fields and who could communicate with different, basically, scientists and specialists from different fields.
Yes.
I'm interested in reflecting back on your postdoc period and then transitioning to basically professorship or having your own lab. How did that experience basically go?
That's always a very delicate step in the career. When you do a PhD, you're still referred to as a PhD student. You're still forming yourself. When you start to a postdoc, you have to start to think of the next step, what you want to do. Do I want to remain in academia? Do I want to go to another type of career within the academic environment or in a company? And so there are so many decisions you have to make, and you start to have also different responsibilities in the lab as a postdoc compared to when you're a PhD student thinking at your project. And you have to think about the next steps in career, yes. So I had a postdoc in Dublin for quite many years. It was a beautiful experience. It was a very large group. My mentor there, my supervisors, had started a new center that was fully dedicated to study how, it was called the Center for Bio-Nano Interactions, it's still active. And that was really about understanding how nanomaterials, nanotechnology interacts with biology, nanobio. And so again, the interface between different disciplines. And but yes, so there I was involved in many different projects within the group, and it was a very beautiful, very intense experience. But then eventually I was also looking for an opportunity to establish my own group. And I was lucky that I was informed of this possibility of the Rosalind Franklin Fellowship that were offered here in Groningen University. These are positions that are specifically aimed at establish female scientists to start their own group. And the beauty of it is also that you can apply with your research topic. And so sometimes you have positions where a specific candidate with a certain profile is looked for, and you have to see if your expertise fits with that. But these positions are, if you want, a bit more open, so that you say, okay, this is the research I want to do, and you see if you find a match and an opportunity in the university. And I was lucky to have that opportunity, and for me it was the time to also start an independent lab, and the challenge there becomes different. You have different responsibilities. The teaching started, my formation as a teacher started, and the work changes a lot as well. When you're a researcher as a PhD student, already when you're a researcher as a postdoc, and when you become a supervisor, a principal investigator responsible of the project of younger colleagues and things like that, it's really the job changes are also quite a lot in these different steps. And the switch between a temporary position as a postdoc and having a permanent position as an independent PI, it's a big one. And it definitely changed a lot.
I can imagine. Academia is known to be a very rigorous career path, especially that you have to kind of establish yourself within your field. But what is really the required step to do in order to become a professor or have your own lab, or to make this transition? To make yourself established in the field, really. Actually,
here in Groningen University, I was also asked to be an ambassador for the postdoc of the Faculty of Science, because, in fact, the faculty recognized that the postdoc step in the career is a very delicate one. You have to decide what you want to do next, and if you want to remain in academia or switch somewhere else, right? So you have to use all your energy at best to make the most of the postdoc period, depending on what you want to do next. And what's required to enter in academia? I don't know. Well, first of all, a lot of passion. First of all, you really have to like the job. That's true for every job, I would say. But yes, sometimes maybe it's a I don't know, when you are a postdoc, you don't have yet a full vision, perhaps, of what the job becomes when you are an independent PI, what becomes your responsibilities, your duties, your daily life as a group leader. So you need to be aware of what is the direction you are heading to and see what's needed to arrive in there. Definitely you have to like the job because that's always needed for whatever career path. There is always some dose of luck that you have to be in the right moment at the right place. It's a very delicate step, and there are a lot of factors to take into consideration. You need to show that it depends. When you are interviewed for a position as an independent group leader, there are different aspects that are considered, whether you're showing already that you have the capacity to lead independently a project, whether you have already experience with applications to get funding for your research, whether you are already you have an established network that people already know you at national or international level. There are many parameters, if you have already or not experience with teaching, when you're applying for a position in academia that also involves teaching. So there are many different factors that are important and that are considered when selecting a new group leader.
Yeah, because I was thinking specifically, for example, let's think that someone is applying for or aspiring to become an assistant professor after his post or her postdoc. Is it only the number of publications and the impact of publications, or are there any other features that are being looked for in academia?
No, there are a lot of things. And also the way we evaluate research outputs is also changing. And there is a lot of discussions of how much to count the number of the papers, the impact factor of the papers. But so also this criteria for evaluating careers, the criteria are also evolving. And I think in part it's also reflections of the fact that, in fact, there are many aspects that are required that you need to show you are developing to get to establish your independent group. And it's not only the number of papers and the impact of the papers. Sometimes that might be affected by the topic of the research. So some topics might be more have the possibility to be published in very different type of journals with different impact. So the impact factor of the publication of the journal in which your work is published doesn't necessarily is not the only criteria that one should consider. And for a person to start an independent lab, you look definitely at the research outputs, but also science, the experience in supervisions of younger colleagues, in experience in teaching, experience in acquiring funding for supporting your research, the network. All these other aspects also matter, and you have to show how developed you are also in all these other aspects.
Interesting. So it's also a lot of character development and the leadership, basically, skills of the group.
The leadership and management skills, yes.
Interesting.
Yes, absolutely. It's not only how many papers and research outputs.
Fantastic. That was a lovely break. We would like to move back into the theory. We've covered a nice kind of background of how to become a researcher and what it takes and what you need. But yes, we would like to start with an elementary question that can become quite complex once we dig into it. What do we define as a nanoparticle? And I guess what makes them interesting in drug delivery? Start from there. Okay.
Well, actually, it might seem a simple question, but there are projects that have been dedicated in the definition of nanoparticle and nanomaterials also because they are consequences, for instance, for regulatory level and things like that. In the context of drug delivery and nanomedicines, there have been also similar debates on the definition. Nanomedicines, there are really many definitions, but you can say that a nanomedicine is a material that has at least one dimension in the nanoscale size.
Okay.
So, but typically, maybe it's easier to think at typical nanomedicine, typically drug delivery systems as a reference. So I was mentioning the first nanomedicine that was approved was a liposome, lipid vesicles. So the lipids have a bilayer similar to the cell membrane. And typically the size of these vesicles is 100 nanometer. So that's a good reference. In general, we're talking of objects for drug delivery, we use objects within 10 nanometers or slightly less. There are some ultra small ones that really you can arrive to 5 nanometers. Then you start to have the size comparable to large proteins. So consider, a large globular protein might have 5-6 nanometer diameters. So the smallest nanoparticles that we might use in drug delivery have a size comparable to a proteins, but typically we use larger object up to 100, 200 nanometers, but it can go also a bit higher up. That we use, in fact, as a transporters of drug molecules. So we put a lot of drugs inside these objects to then help transporting the drug into the body.
So is the nanoparticle the carrier, or is it the carrier plus the inside?
Typically, the drug delivery system is typically a nanoparticle. And then we speak about the load of the drug carrier, as the drug is the load that is transported by the nanoparticle, by the drug carrier. Yeah. So you have a drug delivery system that is typically a nano-sized material, a nanoparticle, in which there are drug molecules, active ingredients, and then the nanoparticle, the nanomedicine, is there to help the transport of that molecules in the body.
Yeah. So in that sense, how is, for example, a paracetamol tablet different from a nanoparticle tablet or any
then we are talking about
medicine, for example?
Yes, no, of course. A tablet, like the medicines that we have at home, tablets are bigger, right? But what we're comparing is the active ingredient alone, paracetamol molecule. So that one is perhaps really 1 nanometer or so, if not smaller. And the difference is that instead of just administering paracetamol molecule as it is, we first load the molecule into a larger drug carrier, 100 nanometer size, for instance, so that one of these objects, one of these nanoparticles, contains a lot of your molecules of drug inside. And currently nanomedicines are typically administered by injections, most of them. So we don't have yet tablets made of nanomedicines. So the tablets you're referring to, we don't have nanomedicines that are that are already administered orally, for instance. Most of the nanomedicines are applied by injections, intravenous injections. The COVID vaccines were intramuscular injections, right? That's because in general, for a drug to arrive to the place where it has to do its action, there are many barriers that needs to be crossed. So nanomedicine, it's a technology to help the drugs transporting it through the body and through these barriers. But it's still quite challenging. If you immediately inject in the blood, you have crossed a lot of barriers already, right? You're already in the systemic circulations, and your nanomedicine carrying the drug is already in blood. For oral administrations, you first have to formulate your drug into a pill or some other form, and then you have to, the medicine has to transport, has to arrive at the gastrointestinal level for being absorbed and everything. And nanomedicines at the moment are mainly applied by injections. So if you go with oral administration, there are other barriers that needs to be crossed, and typically we don't have yet nanomedicines that can do that. It's under research.
Interesting.
Coming back to the matter of definition of nanomedicine.
Yes.
So can I maybe summarize it, if I say it correctly, that nanomedicine, the fundamental unit in nanomedicines, is not the molecule anymore, but it's the carrier which can contain many molecules.
Yes.
So
Correct.
Is it scaling up or is scaling down in this case then?
Well, for me, it's a scaling up. So your active, perhaps when we have a drug that we administer orally so when you think at a pill, it looks like you go from a big pill to a small nanomedicines. But think instead, perhaps let's take the sample of chemotherapy. So for cancer treatment, typically chemotherapies are administered by injection in blood, right? And those chemotherapeutic agents are small molecules, doxorubicin that I was mentioning before, that have perhaps just a nanometer in size or so. So these are medicines that we administer in blood. But then the way they distribute in the body into many different places, and they cause a lot of side effect. So nanomedicine there is try to avoid the distributions of target distributions in all the organs that are not intended to be reached. And this technology to help you to deliver the drug, in this example, only at the tumor site. So by encapsulating, by loading your chemotherapeutic molecule into a nanomedicine, into a large drug carrier like a liposomes of 100 nanometer, you put a lot of the doxorubicin molecules inside this drug carrier. And when you inject that, now the way that nanomedicines transport in the body is totally different than the molecule alone. Because you have passed from doxorubicin as a small molecules that can cross through cell membranes and partition in the body and can reach many different organs, including the tumor. But when you have 100 nanometer liposomes carrying a lot of doxorubicin molecules, it can only enter by active mechanism of uptake. It cannot diffuse through the cell membrane. And also, often for cancer therapy, nanomedicine take advantage of a phenomenon that is called enhanced permeation and retention effect that we use for passive targeting of nanomedicines, meaning many tumors have blood vessels that are a bit abnormal comparing to healthy blood vessels. Typically, blood vessels are, you have endothelial cells, the cells of the blood vessels that are tightly connected to each other to form a barrier so that we control very carefully what goes from the blood into every tissue. But tumors sometimes grow very fast. They need the blood vessels to support the cells, the tumor cells. But the way these tumors form, the blood vessels form, is abnormal comparing to an healthy tissue. And the blood vessels at the tumor site, instead of having a perfect tight barrier, might have leaky vasculature. The vasculature at the tumor site can be leaky. And nanomedicine takes advantage of that because if you have 100 nanometer liposomes, now it can passively extravasate from the blood into the tumors through these gaps that the tumors has into this because of these abnormalities. So if you inject doxorubicin, you have distribution in many different organs and the passive crossing of the cell membrane. The molecule is small, is not charged, and can penetrate into cells. It accumulates in, for instance, it has a very strong cardiotoxicity, neurotoxicity, and also there are a lot of side effects that are due to the fact that we have a poor delivery. We don't control where the drug molecule goes, and this molecule is kills cells. Not only tumor cells, but whatever cells it goes, essentially. So if it's not delivered at the tumor, but it goes to the cardiomyocyte, the cells of the heart, it can kill those cells. And it's the same in many other different locations, and those are the side effect of chemotherapy, right? I think we're all familiar with that. Encapsulating the doxorubicin into the liposomes prevents these passive diffusions to many different organs, and helps the drug stays longer in blood because small molecules can filtrate to the kidney, exit the body very fast, while 100 nanometer liposomes cannot be filtrated by the kidney, stays longer in blood. If you stays longer in blood, you can have, in the circulations, you have longer time to cross the next barrier, in this case to go from the blood to the tumor tissue. And because you have these leaky blood vessels, you have passive distributions from the blood into the tumor tissue. And actually, there is also typically the nanomedicine remains there also for longer because tumor tissues might lack of lymphatic systems. So you have an enhanced permeation and an enhanced retentions at the tumor side. But going back to the basic of nanomedicine, many nanomedicines are among the approved ones are also drug delivery systems that allow you to completely change the way the drugs distribute and move into the body. By encapsulating small molecular drugs into a nanomedicine, you prolong the time the medicines remain in blood and circulate, and you improve distributions, for instance, towards the tumor via passive targeting, and you reduce these passive accumulations in many other organs where you can have side effect. So in general, what nanomedicine tries to do is controlling better how the drugs controlling better where the drugs goes, and provides you a technology to push your drug towards the targeted side.
So that's very interesting that we can minimize the side effects and have
better delivery to
better
the tumor
delivery.
in this example.
But what is really the magic here? What is the encapsulation? What is the carrier? Why is it so effective?
Well, the size is the size that changes a lot the way the object distributes the pharmacokinetic and the pharmacodynamic, if you want to use some language from our courses. So the fact that you encapsulate the molecule into a nanomedicine and to a larger objects now, as I was saying, changes completely the way this medicine then distributes in the body and how it's transported in the body.
Is it now also the composition? Because I think Oscar had also a question about that. Maybe you could formulate it better than me.
Exactly. Like we mentioned liposomes. Is that the only? So are we talking about a lipid bilayer specifically that encapsulates most of these basically drugs that we embed or within this kind of lipid bilayer, or is it more complex than that?
There are many different types of nanomaterials that we can use for nanomedicine. So liposomes were the first that were approved, and there are many nanomedicines that are liposome formulations. But you can have, in general, there are lipid-based drug carriers, so you use lipids and the fact that lipids have assembled into different structures to help the transport of drugs. The COVID vaccines, for instance, are also lipid-based drug carriers, but instead of liposomes, meaning a lipid bilayer with inside some aqueous environment, you have the vest is a full it's a fully lipidic vesicle. So also the inner volume is full of lipid molecules. Okay? And it's a solid lipid nanoparticles, it's called. So you have many different types of lipid-based drug carriers, but you have also a lot of polymers that we use for drug delivery, and it goes, the amount of material and the type of design that you can have is really, really huge and vast. You can have inorganic materials that we use for drug delivery. They also provide inorganic materials, like gold nanoparticles, silver nanoparticles, silicon dioxide. They're material that provide additional properties. For instance, they allow to do new imaging modalities so that you can also see the nanomedicines and image where the nanomedicine goes. Sometimes inorganic materials allow new forms of therapy. Instead of delivering a drug, you can have inorganic nanoparticles. Some of these materials can have the capacity of absorb light or interact with magnetic field or with laser light and respond to that. They get excited in different ways and they can relax their energy, generating heat, essentially. So there are many interesting applications of inorganic nanomaterial for thermal therapy. You take advantage of the capacity of this material to absorb light and get excited and then release their energy, generating heat locally as a new form of therapy. So because the heat that is generated can be used itself to kill cells, or it can be used, temperature increase can trigger release of a drug from a system that responds to temperature. So with inorganic materials, we can also make what is called a stimuli-responsive drug carrier that release the drug only in the presence of a certain stimulus that could be the laser that you apply to the patients so that you can really control where and when you apply the lasers, and only in that spot at that moment of time your nanomedicine activates a therapy based on thermal effect or release the drug that is transported. So there are beautiful possibilities within organic materials, but not only. In general, you can also engineer nanomedicines that respond to stimuli. And that's a way of controlling, again, where the drug is released and has its action. And so lipid-based, polymer-based, inorganic materials, and also a lot of bio-inspired material. Our body also has nanoparticle to deliver compounds in the body. Think how we transport cholesterol in the body. Cholesterol is a very hydrophobic molecule, so to transport it in these different organs, like the chemotherapies also are androphobic compounds. You need a nanoparticle to be able to improve the solubility of cholesterol and to transport it in different organs. So we have what we call the low-density lipoproteins, high-density lipoproteins. Those are all nanoparticles, essentially, because they have sizes within 20 and 100 nanometers, where a poorly soluble compound, cholesterol, is packaged with proteins that help solubility in blood. So you have lipid nanoparticles to transport poorly soluble lipids into different organs. So that's an example of a natural nanoparticle, if you want. And then we have also exosomes that are vesicles that are released by cells for communications to other cells, and not only. So exosomes are also lipid bilayers with a lot of proteins and very interesting cargo, very interesting load. And again, those are lipid vesicles of around 100 nanometers, not so different from the liposomes that we use in nanomedicine, right? So viruses, of course, that's the top of the nanoparticles. Viruses, we also use viruses for drug delivery, in fact, because they are unique particles that have the capacity of delivering DNA into the nucleus and things that we perhaps don't manage yet with nanomedicine.
So we use viruses as carriers?
Yes,
as
absolutely.
Carriers of the nanoparticle.
Yeah. So, for instance, again,
is virus no bigger than the nanoparticle, nano size, or
you use the virus as a carrier.
As a carrier, but
yes,
it's still bigger than nano, or not? Or is it
comparable sizes.
Comparable, okay.
Also, again, some viruses within, I don't know, depending on the type of viruses, but 100 nanometer is a typical size of many types of viruses. So what we do is we change the genetic content. Think about the DNA vaccines for COVID-19. You have a viral nanoparticle, a viral particle in which we have delivered the DNA molecules that is producing the spike proteins, right, for the vaccination. So perhaps we don't refer to this as nanomedicine, but that's again a natural kind of nanoparticle, a virus that we use to transport, in this case, a DNA piece into specific location. So there are a lot of bio-inspired nanoparticles. We try to learn how lipid nanoparticle, viruses, exosomes are trafficked in the body and they distribute, how they target the cells where they need to go, so that we can get new ideas and new strategy that we can use in drug delivery as well.
Yeah. Okay. Well, it's very creative to use viruses. Now I'm thinking about it. You can get the virus just to infect the cell, and then to produce the protein or the therapeutic protein.
Yeah,
and
viral vectors have been, viral gene delivery and viral vectors have been studied again for a long time. And again, they are so unique, and being able to deliver DNA in the nucleus, for instance, that we try to exploit that capacity for gene delivery applications. Yeah.
Okay.
Now we have these different surfaces of the nanoparticle carriers. We inject it in the blood, and then we get a lot of protein accumulation called corona.
Yes.
Does the surface matter anymore? Because now we have the protein as the surface.
Absolutely.
Maybe if you could just reflect on the idea of corona protein.
And to quickly specify, this was specifically with lipid bilayer-based ones, so kind of liposome.
Or was
type,
it also with organic?
Exactly.
It's a general phenomenon.
Okay,
it's a general phenomenon. So if you have nanoparticles, the surface of the nanoparticles is very reactive. It sticks a lot. to anything. So if you inject nanomedicines in blood, it doesn't matter if it's a liposome, a polymer, an inorganic material, it tends to absorb molecule on the surface. And well, we also know, I mean, that's a very general phenomenon. It's true also if you have biomaterials that we use for implants in the body. The implants also bind proteins, right, and molecules. So what is unique about this corona that forms on nanoparticles is that the composition of this layer is quite unique. It depends on the property of the nanomaterial, but it has a very specific composition. So when you inject nanomedicine in blood, we think about unspecific bindings of proteins, right? But instead, what we have learned in the last decades of research, the name protein corona was introduced in 2007, so it's relatively
before
recent.
the epidemic, actually, yeah.
The phenomenon, it was well known. What was not known is that the implication then for the behavior of the nanomedicine or the nanoparticle in the body. It's not really just unspecific binding. Some proteins that are very low in abundance in blood can really attach very strongly on these particles. And it is true that in the end, we have more and more evidence that in the end, the cells interact with this layer of molecules absorbed on the surface that can hide the nanomaterial that is underneath it. So sometimes this can be a problem because perhaps for nanomedicine, for instance, we attach on the surface of nanomedicine antibodies or ligands that we want to be specifically interacting with the targeted cells. So we modify the surface of nanomedicine with ligands that should recognize the cells that you want to deliver to. But if then now, once you put it in blood, a lot of molecules absorb, those ligands might be covered. So you have to design the material knowing that you will apply it in blood, and you have to counteract or expose this your targeting ligand even when in blood many molecules might absorb on your particle.
Okay.
And actually what is coming, what more recently it has been discovered, not only work in our lab, this is really something that is changing a bit the field, that we have more and more evidence that some of the molecules absorbs interact with receptors. And we have a nanomedicine that has been already approved in this use in the clinics for a rare genetic disease in the liver. This nanomedicine is called Onpatro. It was approved only in 2018, quite recently, a few years before the COVID vaccines. And it's the same technology of the COVID vaccines, actually, because it's a lipid nanoparticle to deliver RNA, but the type of RNA is a bit different. And anyway, it was discovered that these nanoparticles, this lipid nanoparticle, goes to the liver, not because it has an antibody on the surface to target the liver, but because once you put it in blood, it absorbs a very specific corona, and it is a corona particularly rich in proteins that interact with specific receptor of the liver hepatocytes. So it arrives in the liver, into the cells in the liver hepatocytes because of the absorbed proteins in the corona and the presence of proteins that can interact with specific receptors on the hepatocytes.
But did that accidentally work? Because
Absolutely, yes.
that other proteins would adhere to the corona or to the
This was something that it was not intentionally done so for that effect. It was discovered that these particles had the capacity to arrive to the liver hepatocytes. Many nanomedicines end up in the liver. That could be a problem if you need to go somewhere else. But typically go to the macrophages, to the Kupffer cells, the phagocytic cells of the liver. This particle instead is going to the hepatocytes, so different type of cells within the liver. And the people involved in the development of these medicines have understood that this targeting to the hepatocytes is provided by these molecules that absorb in the corona once you administer it. So this was, something understood afterwards. But then as this corona field has been increasing and more we understood the unique characteristic of this layer and the fact that it can be so specific, the more we have seen evidence, we have example also from my lab where we see that specific corona proteins can interact with specific receptors. So the receptor is mediated by, the uptake of the nanomedicine is mediated by an interaction of a corona proteins with cell receptors. We have more and more evidence of this kind of phenomena mediated by these adsorbed proteins. So now that we know it, people are trying to say, okay, can we use it? So what many people are trying to do is prepare a lot of different type of particles. If you change the size, the charge, the material of the nanoparticles, what adsorbs on the surface is different once you put it in blood. And people are trying to use this to discover corona that drives you to other organs. So, but it is a bit of a trial and error, unfortunately, because you screen libraries of particles, and then you see if it goes in a different organ, and then they study what is it on the surface, and you try to discover if it is driven by specific proteins that adsorb on the surface only when you administer it in blood.
I'm curious, is it a chaotic process, or is there some kind of pattern that we could discover and exploit?
Yeah, well, it's not chaotic, but it's a very complex mix of interactions. So there are also people that are trying to understand exactly can we predict which corona forms, what are the rules that govern corona formations, so that we can better exploit it, better control it, better avoid it when we don't need it or it affects the outcomes in a negative way. So it's a topic of research itself, and what we understood is that it's a phenomenon different than what we normally observe with biomaterials, with implants that also stick proteins. So the fact that you have a nanoparticle, highly curved, small object, changes the nature of what adsorbs on their surface, and there is a lot of research in understanding the rules so that we can control it and use it. Absolutely.
Isn't this a little bit paradoxical? Because the whole idea of nanoparticles was to have a controlled delivery system, and now we know that there's this uncontrolled pattern of proteins that would change the fate of the corona particle or the nanoparticle practically.
Well, we need, I think it's, we have to not exaggerate, we have to still understand a lot about this, and is not as chaotic and uncontrolled as it might sound when you think about it. So we can reproduce it. We are learning the rules about it and how it matters. We still need to understand to what extent it matters and how much it affects the interactions. So now it's in a moment where perhaps these recent findings are creating a lot of excitement about the possibility of using it in a positive way. Typically, you think about trying to decrease protein binding so that your targeting works. Now that it's appearing that some of the proteins instead might provide targeting themselves, there is a lot of excitement in the possibility of using it. We will have to do more research to understand how much is usable, to what extent, and how to control it.
And personally, what is your preference? Would you prefer to, for example, embed proteins within this nanoparticle or nanodrug carrier to maybe repel basically the protein corona kind of formation, or like to influence it in a specific way? What is your school of thought? And maybe
there are a lot of interesting possibilities. So one thing is indeed that you can. Definitely, we can design nanomedicines that have less tendency of absorbing proteins and molecules. So we can make surfaces of nanomaterials less sticky to control and try to diminish this kind of effect. The problem is that if you make a nanomedicine which really doesn't stick any proteins, that really interacts very little, there is the chance that it doesn't even interact with your cells and with your
Exactly.
target. So it's always a matter of balance between the interactions that you want to promote and the one that you want to avoid. And that's the chemistry challenge in how to design that. So if you make the most repellent surface, it will also not enter cells. So it's really about how to design it in such a way that you achieve the ones that you want, the interactions that you need for your delivery, for your targeting, and not the one that instead block it and drive it somewhere else.
Of course.
So we have many chemistries to possibility to design surfaces that are less sticky to reduce the phenomenon. But then you have to balance it with the fact that you want also some interaction. You cannot only shut down every interaction, otherwise you will never enter any cell. You will not deliver. I think we need to study more the mechanism behind it to see to what extent we might use it. But I think we also need to learn. I would not abandon targeting strategies where we try to modify the surface to attach ligands that help that drive the distribution to the targeted cells. I would not abandon that kind of research either. The important thing is that we are aware that once you apply it in blood, it might completely change. We need to be aware of this phenomenon that once you apply it in blood, it might change completely the way it interacts because of these adsorbed molecules. We cannot ignore it.
Yeah. But does that mean, for example, that if you would administer it via the blood, it would be different than if you administer it via the lungs?
Absolutely.
Then you change the nanoparticle based on the administration route.
You change the corona that absorbs depending if you inhale nanomedicines or inject it. If you want, we can make it more complicated than that.
Even more complicated?
But again, I don't think we need, what we have to do with the research is not get lost in this complexity. Now we are opening up this complexity, and then we need to distill what matters and what not. But what has been observed, for instance, in a work I did together with AstraZeneca, we compared the corona that forms in lean and obese plasma.
Oh, interesting.
So certain the physiological status of the individuals or the diseases can affect, for instance, serum plasma composition. So it's no surprise then that because you mix your nanomedicine with a different type of plasma, the corona that forms is different. So that already tells you that, yes, a lean and an obese patient might end up with a different coronas. And you can multiply it, and some people define it as a personalized corona, in fact. Again, we need to understand to what extent it matters, right?
And what things can stay constant and what things will be...
but the observations in the studies that we did, that mainly people in AstraZeneca did, that was a collaborator, was very interesting. The lipid nanoparticles, similar to the COVID vaccines, but in lean and obese plasma, they form different corona, and then the transfection efficiency, most likely the amount of RNA that is delivered in the cytosols was different. So it really affected the
actual efficacy as well.
The actual efficacy in this case.
Yeah.
Now, this was done at the cellular level. But again, is the concept that the important concept is, again, that the nature of the environment in which you apply the nanomedicine can change the biological activity and the distributions, the receptors interactions. So in this moment, the list is getting longer. I think we have to understand better, also to not get lost in this complexity, to make a sense out of this complexity, see what are opportunities and what are the problems.
Exactly, that was my question. So,
yes.
Do you still see? Do you still think that in the near future we could make practically effective nanoparticles, even if we lack the understanding of how nanoparticles would adhere to different proteins in the body, or is that kind of counteracting the efforts of, for effective nanoparticles?
Well, we definitely have many nanomedicines that are already approved and are helping us.
Even if we don't understand them properly.
there are some aspects of the mechanism behind that still need to be elucidated. That's the true also for molecular drugs, not only for, not always only for nanomedicines, absolutely. So now with this topic specifically of the corona that absorbs some nanomedicines, we need to understand the opportunities and the problems and the level of complexity, so that the important thing is that we're aware of this phenomenon, but we still have to do more research to really appreciate what it means, to what level we need to be able to control it, right?
Yeah. And I have actually, the question might be answered by the fact that it is a lot of endogenous proteins that absorb to the
Yes.
lipid carrier to begin with. But is there any issues with immunogenicity sometimes with these particles, or have we experienced such things? Or is it because these endogenous proteins cover the surface and basically make it
Mimic it.
mimic the
Yeah.
as in a particle of self, I guess.
Again, there have been different observations and different contexts in which maybe both aspects seems to be present.
Okay.
So, and actually, you mentioned these are endogenous proteins. Some people refer nowadays to this corona-mediated interaction as endogenous targeting, in fact, because it's a targeting capacity acquired by the nanomedicine, is not passive targeting. The leakiness through the vasculature of the cancer cells, the cancer tissue, is not active targeting. You put an antibody on the surface that specifically recognizes the target cells. It's an endogenous targeting acquired only once you put it in blood and molecules absorb on the surface, right? But sorry, now I think I've lost the track of my of the thoughts.
No worries.
Your questions was
immunogenicity.
Immunogenicity.
Yeah.
Oh, okay. So one nice thing that was some findings challenge a bit this. For instance, it's a bit of a complicated story. One of the ways to reduce protein binding on nanomedicines or on surfaces in general is to attach a polymer on the surface that is called polyethylene glycol. You attach a polymer that makes the surface less sticky. And typically what we think about how it works is that because it sticks less protein, these proteins can be signal for cells of the immune systems to remove the particle. We talk about opsonin proteins that absorbs on a surface and are a signal for macrophages to clear the particles from blood. So if you attach these polymers that reduce stickiness, you stick less proteins, you stay longer in blood. That's the observations. And we always thought we stay longer in blood because we reduce this protein binding that activates removal of the particle. Okay? Actually, some people have shown that the prolonged circulation times is not because there is less protein sticking, but because there are specific proteins in the corona that forms also on this type of surfaces, and specific proteins that don't eat me signal, let's say, for the immune systems, signal to immune cells, don't eat me. So we also discovered certain proteins that once absorbed on nanomedicines act as a don't eat me signal for the immune system, so you avoid clearance. So those are proteins that you might want to have on your surface. So you can think about manipulating the corona or form an artificial corona before administration with these don't eat me signals proteins. So in this case, it's avoidance of immune cells, if you want. In some cases, there has been examples in which proteins absorbing on the surface can instead activate the immune cells. So understanding whether there is immunogenicity mediated by this, it's also a topic of research. In some cases, it has been found upon absorption of some molecules, these proteins, when they absorb on nanoparticles, can lose their structure partially and expose some new sequences that normally are not exposed outside by these proteins, and that can activate immune cells for clearance. There are also studies that are addressing whether there is immunogenicity activated by nanomedicines in general, and perhaps immunogenicity via adsorbed proteins that might activate the immune system.
Very good.
That's very interesting that you also mentioned clearance.
Yes.
That also means that maybe we could modulate and differentiate the clearance based on the state of sickness. For example, if we have inflammation, or if we have a patient who has different proteins than healthy individual, that means that we could maybe make use of these proteins so that the the nanoparticles could stay longer in the blood than in the healthy individual.
There are a lot of studies now so we have a lot of new research methods to characterize this layer that forms and identify which molecules are absorbing there and how they are oriented on the surface, because not all of them will interact with receptor necessarily. There will be only some outer layer that might have the capacity of interact. Not just because you are absorbing the corona, it means that you interact with the receptors, the corresponding receptors. Absolutely not. It's more complex, again. But so there have been studies where we try to correlate the presence of, we correlate the composition of the corona with increased or decreased uptake, prolonged or reduced circulation time. And in that way, we can identify what you say, we can identify if certain proteins prolong residence time, prolong the circulation times, or others instead reduce it. If some proteins promote uptake by cells and specific cells that you want to target. So there is a lot of studies now, we do it also in our lab, to try to use this corona to discover proteins that might promote or reduce uptake.
Interesting. So the drugs are becoming more dynamic, actually, and individualized to the patient.
Yes.
Are not the same static molecules that will interact the same for every individual and for every situation.
Yeah, actually, you were mentioning before how complex this is and how can we apply to many different patients, for instance, right? But in fact, I think in many other fields, we are recognizing that drugs act in different way in different individuals, right? So there is personalized medicines in the sense that there is best treatment for every patient, but also that the efficacy of certain treatment can vary between patients. And this is not only for nanomedicines, but in the case of nanomedicines, it might be connected to, there might be an influence of these corona-related interactions that changes the efficacy of medicines in different patients, absolutely.
Interesting.
So in general, personalized medicine is also
connected to the nanoparticle realm.
It's relevant here. It's a field of research that it's growing quite a lot because we're understanding more and more the complexity of this. And this is nanomedicine fit into that picture as well. These aspects that we are discussing also suggest a personalized medicines approaches that the same medicines might have different effect in different individuals because of these subtle differences that we need to discover.
Perfect. And so for our final section, we would like to actually maybe even, I guess it's a new segment that we're introducing here, go over some of the research techniques that on a daily basis maybe a PhD student or a postdoc uses to actually test nanoparticles, perhaps these corona protein formations that we discussed at length in the previous section. What techniques do you use? Like just broadly, I guess, and then we can zone in on some of them.
Okay, so our lab is mainly focusing on cellular work. So we culture cells, we grow cells, sometimes not only simple cultures, but more complex cellular barrier that mimic endothelial barrier, or we've done some work also with tissue slices that we have as a technology in our institute. But anyway, we mainly do cellular work. We don't do animal work. And also we do, so a lot of our work, it's about adding our nanomedicines to the cells. First, we put the nanomedicines in relevant biological fluids, in plasma, in serum, and we study what happens in there and all this corona-related characterization. And then we study the interactions with the cells. There is a lot of cellular work. And typically we have fluorescently labeled nanomedicines that we use. And we use fluorescent-based methods like flow cytometry and fluorescence microscopy to quantify the uptake, see where the particles are inside the cells, and follow the process. So yes, definitely cell work, flow cytometry, fluorescence microscopy, it's our daily activity. And we are not a chemistry lab, but we do a lot of nanomedicines, nanoparticles ourselves. Typically, we work a lot with commercially available materials, but we also work a lot with lipid-based drug carriers. Because the beauty of those, apart that they're very relevant for medicines, because the most approved nanomedicine, the most frequent types of nanomedicines are lipid-based drug carriers. So liposomes and lipid nanoparticles, if you think now at the COVID vaccines. So the RNA vaccines. So we have systems to different methods to prepare liposomes and lipid nanoparticles. And it's a very versatile system. So you can change the lipids. So by that, and you can make different type of lipid-based drug carriers, and by changing lipids, you can change the charge and the surface properties of these different liposomes and lipid-based drug carriers. You have methods to control the size. So it's also very versatile model systems to then interrogate the cells and understand how all of these parameters of the nanomedicine design affect the cellular behavior. The interactions with blood and serum, and the interactions with the cells afterwards. So if we want to do, so some people are more, then again, we go back to interdisciplinarity. We have some students that have more background from physical chemistry, perhaps, and are interested in the formulation, the design of the material. Some people that are more interested in the proteome, in the corona-related research, some more in the cellular mechanism. A lot of what we study is really the cell biology of nanomedicines. How do they enter? Where do they go? Which receptor interact with? What is the mechanism of uptake? Where do they release the drug? This kind of aspect. So really, how the cell works and how nanomedicine interacts with these cells. So with the idea that understanding the mechanism, we know how to design, we understand better how to design the nanomedicines that does what we want the nanomedicine to do. Yeah. That's the idea. So there's really a lot of method in cell biology to study pathways and mechanisms. So, inhibitors, knocking down proteins, productions of fluorescently labeled proteins, all the method in cell biology to characterize cellular pathways that then we use to characterize how nanomedicines interact with and are processed inside the cells, depending on the nanomaterial properties. For corona studies, there are a specific set of methods. Some of these molecules, once you put the nanomedicine in serum, in plasma, for instance, to mimic exposure in blood, there are many different methods, and some we have also optimized and developed ourselves to isolate these corona-coated nanomedicines, removing the ones that are not adsorbed, and then to characterize what is on the surface. And there typically we use proteomics, mass spectrometry, to identify the proteins that are adsorbed on the surface and understand more of this corona. We use proteomics also with the cells to for instance, identify the receptors that interact with the nanomedicines, or to characterize in which intracellular compartment the nanomedicines are brought and trafficked. So those are also methods that we use quite a lot, yeah.
And are these in vitro studies for the corona, or do we need another setup for corona?
We do, since we don't do in vivo work, we typically study the corona by mixing our nanomedicines or nanomedicine models with serum or plasma. And for instance, we also change the concentrations, because when you do cellular study, the serum is just something that we add to the cells for feeding the cells. And typically we use, for instance, think about this as well. We use diluted serum, 10%, typically from bovine serum, because it's cheaper But once you start to think at all these corona-related interactions, then you say, why bovine if I'm studying human cells? And why 10%? In the blood, the serum is not diluted. Plasma contains proteins that in serum have been removed, all the coagulation factors. So even before we go to an animal work, there is so much that we can study, even simply by using relevant biological media and studying this phenomena. It's also true that studies that have really recovered nanomedicine from blood in animals showed that there are differences comparing to what you have outside of the animal, right? Because you are missing cellular proteins secreted, other proteins that are that are secreted from the cells. We can mimic the fluids. We can do dynamic studies. We can expose nanomedicines, mixed nanomedicines in blood in the presence of flow. We have microfluidics to do these kind of things. But of course, there are some aspects that you can study, some aspects of the corona that forms only in the animals, in an alive animal, that are still hard to reproduce ex vivo.
Is this in vivo work still meaningful regarding that the nanoparticle is dependent on the biological matrix?
That's a big question in general for the whole drug development process. How much can we use animal studies? How much they can be translated into humans, right? So there is a lot of research into advanced alternative testing strategy, not only to reduce the use of animals, but also to establish models that are more relevant for the human physiology, for the human situation, for human body, right? So definitely that's also an interesting part of research, and we do a bit of that. I mentioned tissue slices. I had a project in collaboration with Peter Olinka that is working a lot with that technology, and thanks to that we were able to study nanoparticles in real tissue, including human tissue that we received from the university hospital, and that's quite a unique opportunity already to try to use alternative testing strategy and more complex than a simple cell culture. But of course we also did there is already a lot of differences if you grow the cells in a plate in a standard cell culture or if you grow them into a barrier, for instance. I was mentioning most nanomedicine are injected in blood, so one of the first relevant barrier they need to cross to arrive to any organ of interest is the endothelial cell barrier. So we have had projects in which we learned how to grow endothelial cells into an endothelial cell barrier. So with the cells, that's different than just the cell in a plate. In an endothelial cell barrier, you have tight contact between the cells, the expressions of tight junction proteins. The cells becomes polarized, meaning they express different proteins and mechanisms on the apical side and the basolateral side. So we have had projects in which we developed endothelial cell barrier, also using endothelial cells from different organs, because the blood vessels in the kidney, in the lungs, and in the livers have different characteristics, and we're interested in studying this characteristic again for targeting organs.
Very good. So during all this complexity, this beautiful complexity, of course, do you think that we are far in the nanoparticle research, or do we still just scratching the surface? Or do you see the first drug coming, or like the first really good drug coming into the coming years?
You know, a few years ago, also when I was when I teach in a course on nanomedicines. Well, it's fun. It's interesting to see. When I moved to Groningen, I was asked to develop a nanomedicine course, which was not yet embedded in the curriculum. But it was still 10 years ago. It seems also, okay, let's see how interesting it is, how much we can embed it in the curriculum, right? But then, and also in those years, there was a lot of debate on the success of nanomedicines and how successful the technology is. Because, okay, we have 20 nanomedicines or so, but there have been many decades in which the type of nanomedicines were often liposomes, not a lot of new technologies, a debate on how successful these liposomes are. We can discuss for a long time about all of that. But then in 2018, 2020, suddenly some new technology received approval, some new nanomedicine. For instance, in 2018, I mentioned the first lipid nanoparticles for RNA delivery, actually. The particles I was mentioning before for this genetic disease in the liver, that was actually the first RNA-based drugs that was approved. And we are talking about 2018, a few years before the COVID vaccines. And that already brought a lot of wow in the field because suddenly we could use RNA drugs thanks to again, RNA doesn't enter cells and can be easily degraded. It needs to enter the cells, but then escape in the cytosols. So you really need a nanoparticle to be able to, and nanomedicines to be able to do that. And the first RNA-based drugs, the first RNA nanomedicines was approved in 2018 for this genetic disease in the liver, the one where then targeting was provided by the corona molecules. Okay, and then a couple of years later, we were in the pandemic, and the technology was ready. There was already a lipid nanoparticle for RNA delivery, and many companies were already working on mRNA delivery. The first nanomedicines was for the delivery of small interfering RNA, so short RNA, but there was already a lot of work to try to deliver mRNA, and that was a very fortunate moment in that horrible situation of the pandemic to have the technology very close to be ready and finally ready for approval. So essentially, after that, the way nanomedicine is perceived and the potential of nanomedicine has been understood has changed quite drastically because the application of this vaccine worldwide has changed completely. Also how now research is interested in nanomedicine. There has always been interest in nanomedicines. I've always been there since it was called drug delivery during my PhD and master project on liposomes. But I think now it's a moment again in which there is a burst in the interest in this technology. Also because, in many cases we have excellent drugs. The problem is more the delivery, ensuring that the drugs arrive where it needs to go. Or we can now use RNA, we want to use RNA drugs, we want to use biologicals, gene editing, so these are new therapeutic type of modalities. And in many cases, we need the nanomedicines to be able to deliver it and make it happen. So in this moment, I think there is quite a lot of excitement around nanomedicine. But we have to be careful also when it's like this, because then you can also have too high expectations. There is a lot of complexity that we still need to address. Targeting is still quite challenging. So yes, all these technologies are allowing us to deliver RNA, to improve the targeting, active targeting, passive targeting, endogenous targetings, but the success of targeting is still might be enough to achieve certain therapeutic effect, but we need to improve, really be able to target drugs to their site of action. So there is still a lot to do to go farther.
Yeah.
Do you have any further questions on the research?
I know that it's complex, but I guess what is that multi-million idea or next
question,
kind of
the $1 million
question to solve?
Yeah.
What may it be, at least in your opinion, like one specific one, if there is one specific one, but I assume it's...
Targeting is still the big one.
Okay.
So we managed to deliver better at the target, but we're still talking of a very small percentage comparing to what we administered. Mm-hmm. So we still need to be able to improve our technologies for targeting.
And to improve the efficiency as well.
Absolutely.
Yeah.
Yes.
Fantastic.
Yes. It might be via the corona, via an antibody. It might be via lasers and stimuli-responsive materials. Those are all new opportunities for targeting that we need to advance further to really be able to achieve it.
Fantastic. Well, I think that's all we have on our side.
Yeah,
so,
of course, the questions never end.
It's really we could continue for hours, but
I can talk for hours.
Yeah, exactly.
Also.
Well, unfortunately, we don't have the time today. We'd love to, but yeah, this was fascinating. Thank you so much for joining us.
Thank you very much. It was very interesting to discuss.
And I guess,
thank you for the rich content. And,
exactly.
for this dense interview.
And we'll be keeping an eye on this field because it's It's really,
A
really
lot is
interesting.
happening.
Yes, exactly. Fantastic. Thank you for
very much.
listening.
Thank you.